This conference will now be recorded.
Hello everyone. Last day we started the topic disaccharide and only the first disaccharide, which is sucrose, that we have seen. Now two more disaccharides are there, that is example of disaccharide. One is lactose, another one is maltose, that will finish today, and after that there will be polysaccharide and obviously problems related to them. So the second disaccharide here we have is lactose. Okay, as you can see the structure, two monomer units are there. So if you do hydrolysis for lactose, there will be two monomers. So that is why it is example of disaccharide. Now this disaccharide, it is found in the milk of mammals and it is referred to as milk of sugar because this molecule is found in milk. So that is why it is also known as milk of sugar. Now the one monomer unit that you are seeing in the left hand side, that is beta D galactose, and another unit which is in the right hand side, that is beta D glucose. So these are the two units lactose is consist of. Okay, this glycosidic linkage that is present between these two monomer unit, I mean beta D galactose and beta D glucose, these two units, they are joined by 1,4 glycosidic linkage. So this glycosidic linkage is formed between C1 carbon of galactose and C4 carbon of glucose. Now this information, that is the third point, is very important because question regarding glycosidic linkage in different sugar molecules, this type of question is very common. Okay, so it is 1,4 glycosidic linkage. This much information is not enough. You also have to remember whether it is alpha or beta. So it is beta, and when it is alpha and beta, there is no direct way based on which you can decide. Actually, you have to simply memorize it. Okay, when stereoisomer of these two carbon that are actually joined by this oxygen linkage, their stereochemistry is different, then only it is beta. If same, it is alpha. So in this case it is beta, and carbon number 1 of galactose monomer unit, it is joined by this beta glycosidic linkage to the 4 carbon of glucose unit. Okay, some more information here, whether it will be a reducing sugar or not. How to know? There must be presence of hemiacetal. So just check in this disaccharide molecule whether there is any hemiacetal present or not. Hemiacetal means there must be this type of structure must be there. So one OH, one OR, and this side there will be any other group. So in our case, as it is cyclic, this part is joined. So this carbon, there must be one OR. Sorry, not this part is joined, I mean to say this part is joined. Here if you look at this carbon, this one, that is the right hand side unit, which is beta D glucose, in that part if you focus on this carbon, you will find there is OR. So this is OR, this side there is also OH, then there is another R. So this part is as if R. So this carbon, just compare with this structure, because of presence of this type of hemiacetal, this can be opened and free aldehyde formation is possible. So these details we have already seen in the last class, that why hemiacetals, if it is present, it will be behaving as reducing. What is the exact reason? Detailed explanation we have seen. Now you can see this example where hemiacetal group is present at C1 of glucose unit, and this unit is not involved in the glycosidic linkage. Carbon number 4 of glucose is involved in glycosidic linkage, not this one. So that is the reason, as this OH is free and that is hemiacetal is present, so that is why lactose will be reducing sugar, which is that is unlike that you have seen in case of sucrose, which is another example of disaccharide. The first example of disaccharide that we have seen, sucrose, you will not find any hemiacetal there. So that is why that example, I mean sucrose is not reducing, but this one is reducing because of this OH. Okay. Now the next disaccharide, total 3 disaccharide. One is sucrose that we have discussed in last class. Second one lactose, we have just finished. And the third one is maltose. Now this is known as maltose because it is extracted from malt. So that is why the name is such. Maltose is produced during digestion. That is, if we consider in vivo reaction inside the body, how it is produced. So it is produced during digestion of starch by the enzyme α-amylase. Starch, what is its structure, its properties, that we will also see in today's class. Right now, just remember, maltose is produced during digestion from starch by the enzyme. Enzyme means biological catalyst, and that is known as α-amylase. So here also 2 monomers are present, and it has to be because we are discussing disaccharides. So obviously there must be 2 monomers. So if you do hydrolysis for maltose, you will get 2 monosaccharides. Okay, so that is why it is disaccharide. Now here also we have 1,4 linkage, right? 1 carbon of 1 monomer unit, 4 carbon of another monomer unit. So in maltose, C1, that is carbon number 1 of 1 alpha D glucose, because both sides you will find alpha D glucose. This is also alpha D glucose. This is also alpha D glucose. C1 of 1 alpha D glucose, that is the first ring structure you can consider, that is connected to carbon number 4 of the second alpha D glucose molecule. And in this case, it is also 1,4, but it is not beta, it is alpha. So in the previous case, you have seen it is beta, but now it is alpha, but it is 1,4. This is also reducing sugar. So just check whether any hemiacetal is present or not. So here also in the same position as you have seen in case of lactose, just focus on this carbon. This is actually the hemiacetal carbon, right? So this OH, as it is free, here free CHO formation is possible. How it is possible? Just like this. If this OH bond is broken, there will be carbon oxygen double bond, and this carbon oxygen bond is broken. So as if there will be formation of OH. Rest of the part I am not drawing, and here we will have C double bond O, which is connected to the other part of the molecule like this. So as by this type of mechanism, free CHO generation possible, or you can say from another point of view that hemiacetal is present. So that is why it will also behave as reducing sugar. It can be produced, that is free CHO can be produced at the carbon number 1 of the second glucose unit. And as we know, this is disaccharide. Both the monosaccharide are same. In the previous example, what you have seen, just remember in case of lactose, you have seen one monosaccharide is galactose, another monosaccharide is glucose. But in this case, both are glucose. Both are alpha D glucose. It is also reducing just like lactose. And if you do hydrolysis in dilute acidic condition, it will give 2 glucose unit because it is disaccharide. Okay. Now we have a question which is based on sucrose. Sucrose we have discussed in last class, and this question is assertion reason type of question. Both that is 2 statements are given here. The first statement is saying, which is assertion, sucrose is a disaccharide and it is non-reducing sugar. The first statement. And the second statement is reason, sucrose involves glycosidic linkage between carbon number 1 of beta glucose and carbon number 2 of alpha fructose. So from these 2 statements, we have to choose the correct option. 4 options are given, right? Now statement number, I'm talking about statement which is in assertion. It is saying sucrose is disaccharide. Now this is obviously true, and it is non-reducing. That is also true because there is no such hemiacetal. Lactose and maltose, they are reduced, but not sucrose. So this is true. Now the reason it is saying that it involves glycosidic linkage between C1 of beta glucose and C2 of alpha fructose. Now if you simply consider the fact that it is C1 and C2, that is fine. But if you simply focus on the fact that one unit is mentioned as beta glucose, another unit is mentioned as alpha fructose. But basically it is true that it consists of two monomers. One is glucose, another one is fructose. But what type of glucose, that we have to see. It is not alpha fructose, okay? That is, so this much error, if it is present, even if other part of this statement is correct, then also as it is mentioned alpha fructose, so it cannot be correct, okay? And if we see the structure exactly, so the first unit that you are seeing, that is actually glucose unit, which is connected by C1 carbon, and the second unit is fructose unit, which is basically 5-membered, and here we have, that is originally, if you consider the structure, it will be like this. So I'm just drawing the OH, which is at position number 2, CH2OH. Okay, rest of the part I'm not. And if you write the glucose unit, which you will obtain after hydrolysis, then if you focus on carbon number 1, then hydrogen, it is, the position is like this, and OH is like this. So this is glucose. I'm just drawing carbon number 1 for glucose unit, carbon number 2 for fructose unit. So glucose unit, carbon number 1, here OH is below, okay? And in case of fructose, which is carbon number 2, here OH is up. So it is true that it is connected by C1, C2. So these two number, that as it is given in reason statement, it is okay. But is it alpha fructose and beta glucose, that we have to check. So basically, it is carbon number 1 of alpha D glucose, not beta D glucose. Okay, so this is not correct. And carbon number 2 for fructose, that is okay, but it is not alpha fructose, it is beta D fructose. If you are having any doubt, I will suggest just go to that slide where I have discussed structure of glucose and fructose individually. There will be different slide. So cyclic structure of fructose, cyclic structure of glucose, which one is alpha, which one is beta, detailed structure you can check there. And now then you will understand that though they are connected by carbon number 1 and carbon number 2, but the glucose unit, it is alpha D glucose, not beta glucose, and the fructose unit is beta, not alpha. Okay, so this is the error. So as it is not correct, so we can choose option 3, which is saying that the first statement is correct but not the second one. Okay, which of the following pairs give positive tolerance test? Now in each option you have two molecules. Both the molecules must be giving positive tolerance test. Then only you can choose it as correct option. If any one of these molecule is giving positive tolerance test, then you cannot choose it. Both the molecules, it should give positive tolerance test. Now the question is, when we get tolerance test positive, obviously tolerance reagent that you use in tolerance test, that is actually mild oxidizing agent. So it means the other molecule with which it will react, it may be sugar molecule or it may be any other organic molecule, not necessarily it has to be sugar, but the other molecule with which tolerance reagent will react, tolerance reagent itself is mild oxidizing agent, so the other molecule that must be having reducing capacity. So any molecule where free CHO group is present, obviously it will give positive tolerance test. Not just CHO, if you have any group like this, suppose you have CH2OH and then carbonyl, so alpha hydroxy carbonyl, not just any alpha hydroxy, terminal alpha hydroxy carbonyl. If you have this type of fragment, then also tolerance test may be positive. Okay, so now one by one we will check these molecules. In the first option there is glucose molecule and sucrose molecule. Now glucose, it can remain in open structure and cyclic structure, and cyclic structure can be converted to open structure also. In open structure there is free CHO, so it should give positive test. But what about sucrose? Because sucrose is disaccharide which is composed of glucose unit and fructose unit. So here we have sucrose structure which is composed of glucose and fructose. Okay, now individually if you see glucose, there is actually hemiacetal present also in fructose, and if you consider their corresponding open structure, in both cases you will find either CHO or alpha hydroxy carbonate. But if you look at sucrose, you will not find any hemiacetal. That means free CHO generation is not possible, so it will not give tolerance test. That is, it will not give positive tolerance test. So it is non-reducing sugar, which we have just discussed some times ago. So it is non-reducing, there is no reducing capacity, so positive tolerance test, that is not possible. But for glucose and fructose, just check these open structures. If you consider the corresponding open structure, so this part, if it is open like this, there will be generation of OH, which is basically this OH, and CH will be generated in this position. So this is responsible for the positive tolerance test. And we also have the structure of fructose. So when it is open, just focus on this part. This is terminal alpha hydroxy C double bond O. So that is why it will also give positive tolerance test. Now option A, it cannot be correct, right? Because sucrose is not positive. This is, I'm giving a cross mark because it will give negative test. This is okay. Now in this case both will give positive. That means this should be the correct option. But we will also check why the other options are not correct. Look at option number D. Fructose, we know it gives positive, but this is not positive, so we cannot choose it. And what about option C, hexanal? Now when it is hexanal, the structure is 6 carbon, 1 2 3 4 5 6, and terminal position there is CHO, so obviously it will give positive test. But the other molecule is acetophenone. Acetophenone is PHCOCH3. Okay, so PHCOCH3 means you do not have any aldehyde, and also you do not have any alpha hydroxy ketone. So this option we cannot choose. Only this one is positive, not this one. So correct option from considering all the logic, correct option should be option B. Okay. Next question. The number of chiral carbon present in sucrose. Now for, that is, sugar molecules, if you get this type of question, chiral carbon, you just have to check the carbon where all the 4 substituents that will be different. So suppose I am writing substituent is A, another substituent B, then C and D. So all the 4 substituents, because valency of carbon is 4, all the 4 substituents that must be different. Then only it will be chiral. Now if we consider the structure of sucrose, you can also check the structure of sucrose. It is already there in the for the previous question, right? So here you can see when it is sucrose, the left hand side unit is glucose, and in glucose 1 2 3 4 5, total 5. But if you consider the other unit, which is 5 member, now there are only 4 carbon that are. So if you consider these carbons where I have given star mark, you will see all the 4 substituents of these carbons are different. So 4 and right hand side, 5 left hand side, total it is 9. It is a integer type of question. So this is the structure with all the chiral centers. Here we have 5, here we have 4. So total it will be 9 chiral carbon. Okay, so these 2 questions, they are based on sucrose. The second one is sucrose, the first one is, there is also mention of glucose and fructose. After finishing disaccharide, next we will see polysaccharide. Now disaccharide means, as the name suggests, when you will do hydrolysis, maximum, that is not maximum, that is each time you will get two monomer, I mean two monosaccharide. But when it is polysaccharide, now after hydrolysis, you will get a large number of monosaccharide units, and all these monosaccharide units, just like disaccharide, as you have seen, glycosidic linkages are present. So in this case also, all these monosaccharide units, they are bonded together by glycosidic linkages, and these polysaccharides, they are the most common form of carbohydrates. Now for polysaccharide, there is no sweet taste present, so you can also call them non-sugar. So sugar and non-sugar, there is a classification. This is a simple classification. If they taste sweet, that is any carbohydrate molecule, if they taste sweet, you can call them sugar. But if they are not, that is their taste are not sweet, it will not be called sugar. They will be called non-sugar. Now this type of monosaccharide, as now the structure is more complex compared to disaccharide. In case of disaccharide, simply two monosaccharides are attached, right? But now there are so many monosaccharides that are attached. So this linkage, it may be linear. That is one possibility, simplest. But another possibility is also there, which is branching. So in some polysaccharide, you can also find branched chain. They mainly act as food storage or structural materials. So food storage, polysaccharide, we will discuss one example, which is for animals, another one for plants. There are three main examples for polysaccharide, and they are starch. So first we will discuss starch, then cellulose, and finally glycogen. So these three polysaccharide examples are there in the syllabus. Starch. Starch is, you have heard this name, it is a very common term. It is used for energy storage purpose in case of plants. So for example, potatoes, corn, wheat, rice, they are very rich source of starch. Now it is a polymer of glucose unit. So here monomer is glucose. And how they are linked? Because that information is very important. It may be 1,2 or it may be 1,4 or it also may be 1,6. So what type it is? It is 1,4. Is it alpha or beta? It is alpha. So this is linked by alpha 1,4 glycosidic bonds. Now starch, there are two main components present. The first component, it is known as amylose. It is water soluble, 20%. That is, if you consider total starch, then 20% of it is amylose, and the rest of the part, 80%, that is amylopectin, which is water insoluble. And when you will see the structure of amylose and amylopectin, this is the very simplest, actually simplest form possible, so that you can understand that amylose is basically linear chain, which is clear from this picture. And there is obviously some spiral nature, as you can see, but this is simply spiral, it is not branching. Okay, so there is spiral nature. This is also important information. If it is asked that among amylose, amylopectin, which one is spiral? So spiral is amylose. It is 20%, it is water soluble, it is linear. But amylopectin, there are some branching. So if you simply focus on this part, this is linear. But see, now from this position, there is some branching. Then again from this position, there is some branching. So now the structure is more complex. It is not so simple like amylose. And this is water insoluble. It is having some branching, and it is 80% of starch. So this is some initial, that is primary information about starch. But now we will individually consider these two part. One is the water soluble part, simple part. Another part is water insoluble part, which is comparatively complex. Amylose. Amylose part of starch. Now as it is part of starch, it is sometimes also called amylose starch. So this spiral structure that you have seen in the last slide, Here also you can see it, coil-like structure. Now suppose if you elaborate this, that is zoom in this small part, you will see there are some units. So these are the monomer units. Linkage is 1,4, only 1,4, and it is alpha. So this is a polysaccharide which is made up of alpha D glucose monomer. So there is only one type of monomer, which is alpha D glucose monomer. How many units are present? This is the limit, 200 to 2000. Greater than 200, lesser than 2000. So that we have already seen, 20 to 30%. Now this is unbranched, that is linear, only linear chain, no branching is there, and it is alpha 1,4 glycosidic bond. Okay, so this is linear, 200 to 2000 units. It is around 20 to 30% of starch, and the linkage is alpha 1,4. So remembering it is 1,4, it is not enough. You also have to remember that it is alpha, it is not beta. Because when you will get question like find the incorrect statement or find the correct statement, then this type of information is very important to answer the question. That is, if it is mentioned that it is beta 1,4 glycosidic linkage, then it is wrong. Though it is mentioned 1,4, but it is not beta. It has to be, that is completely correct statement will be alpha 1,4 glycosidic linkage. Okay, so this is all about the structure of amylose. And if you see the second part, which is around 80%, it is amylopectin. Now in amylopectin, you will see there is not just 1,4 linkage, there is also 1,6 linkage. And just focus on this part. This part is linear, isn't it? This part is also linear, but now look at this. There is branching, right? So as if I draw it in line structure, as if this is a linear part, another linear part, now suddenly there is some branching. Okay, so it is a branched polymer of alpha D glucose subunit. And how many units are there? So obviously now the number of units will be high compared to amylose, because amylose is only 20 to 30%, but now it is 80%. So number of units are also high. I mean number of monomer unit, 2000 to 2 million units, very large, that are linked via the same type of 1,4 glycosidic bond as you have seen in case of amylose. However, there is also alpha 1,6 bond, glycosidic bond, which is not present in case of amylose. So this is important difference between these two. Okay, and 1,6 glycosidic bond is present only at the branching points. So see here, glycosidic bond, which is alpha 1,4, that is present not in branching position. It is in the, when they are in linear form, all these units, individual units, they are linked by 1,4 linkage, not 1,6. But when it is, there is branching, now suddenly there is glycosidic bond, which is alpha 1,6. Now amylopectin, it shows branching at what position? That is also another important information. So it shows branching after 24 to 30 glucose subunits. Okay, and each branch also contains around the same number of glucose subunits. So this is another important information. And this is alpha 1,6, which is at the point of branching only, but 1,4, that is not present in the branching position. Now some question regarding this starch structure. Here we have this question: which one of the following compounds contains beta C1C4 glycosidic linkage? Now in today's class we have already seen all these molecules, right? Lactose, sucrose we have seen in the last class. So lactose, it is example of disaccharide. Then sucrose, that is also example of disaccharide. Then we have maltose, this is also example of disaccharide. These two we have just seen. And amylose, this is a part of starch, and starch is polysaccharide. Now if we consider how to know alpha or beta, though I have mentioned it several times before, but again I'm giving this information that alpha and beta how to understand. An alpha glycosidic bond is formed when both the carbons they have the same stereochemistry, but as stereochemistry details we don't have, that is included in the syllabus, we are not discussing that. You just have to simply remember this information that when the two carbons that are joined their stereochemistry is same, it will be alpha, if not same it will be beta. Okay, so here we have structure of maltose and lactose. Maltose, what type of linkage is present? It is obviously 1,4. So this is 1, this is 4, then this is 1. So you can see the glycosidic bond is between 1 and 4. So that is fine. But is it beta or not, that we have to. So no, it is not beta, it is alpha. Then come to the structure of lactose. Here also it is 4. This is 1. This is again 1. This is also 1, 4. And this is beta. That means this should be the correct answer. But what about sucrose and amylose? Amylose we have just seen. Let's have a look in the amylose structure. See, it is 1,4, but it is alpha. So it cannot be the correct option. And if you see glucose, here we have carbon number 1. This is carbon number 2. So even you don't have to go to that part that whether it is beta or alpha. Here it is connected by 1 and 2. So how it can be correct? So it has to be C1, C4, but it is not C1, C4, right? So this is also not correct. So this should be the correct option. It is beta as well as it is C1, C4. Okay. Assertion reason question for amylose. Amylose is insoluble in water. This is one statement. Amylose is not insoluble in water, right? It is the soluble part of starch. Amylopectin is insoluble. So this is not correct. And what about the other statement? The other statement is saying that amylose is a long linear molecule with more than 200 glucose units. Remember the range, what is mentioned? It is starting from 200 to 2000. And it is mentioned more than 200. That means this statement is correct. So the first statement is not correct. A is not correct, but reason statement is correct. That means correct option should be option D. Next question is for amylopectin, which is the insoluble part of starch. And there is branching present also. Two types of linkages are there, C1, C4, C1, C6. So if you simply consider that fact, amylopectin is composed of C1, C4, C1, C6. That means either option 1, where C1, C4, C1, C6 mentioned, another option where 1416 present is this. But these two cannot be correct because here it is mentioned 14 and 26, but 26 is not present. It is 16 and 14. So 16 and 14, based on this we can cancel option 2 and option 4. Next question is it alpha or is it beta? Okay, so see the same structure as you have seen in the slide where we have discussed amylopectin. Here we have 16 which is alpha and 14 that is also alpha. That means if we consider all these facts, correct option should be the first option, because the other option that is left, these two are already we have cancelled. Other option it is mentioned beta, but it is alpha. Now come to the other polysaccharide. The first polysaccharide example we have finished, which is starch. The second polysaccharide important is cellulose. Cellulose occurs exclusively in plant. It is plant polysaccharide, and it is most abundant organic substance in the plant kingdom also. This is also straight chain. There is no branching. If you do hydrolysis, you will get only beta D glucose. That means here only one type of monomer units are present, monosaccharide or monomer, which is beta D glucose. So It is simple, it is linear. There is only one type of monosaccharide, which is beta D glucose. So after hydrolysis, obviously we will get only one type of monosaccharide. So this is the structure. As you can see in the middle part, we have this third bracket in red color, n number. That means this part will be continued. This is the first position, first unit. This is as if the last unit. Okay. And here we have what type of linkage? 1,4. One monomer, 4 of another monomer. And it is 1,4 fine, but is it alpha or beta? It is beta. So beta 1,4 glycosidic linkage. Where it is found, it is extensively used. That is application, if we consider, extensively used in the manufacture of paper, cellulose, fiber, rayon, etc. And human, we cannot use cellulose as food. Why? Because for digestion of this cellulose, digestion means we have to break all this monomer unit. But to break all this monomer unit in vivo, inside the body, when there is digestion going on, it is not possible, because the necessary enzyme which can break all this glycosidic bond, that is actually not present. So that is why we cannot digest cellulose. Fine. But there is another polysaccharide which is found in animal. So the cellulose polysaccharide, it is plant polysaccharide, but glycogen is, this is also storage polysaccharide, but it is for animals. It consists of entirely glucose molecules. But now, just like previous case, here also we have only glucose molecules. But what type of glycosidic linkage is present? It is now two types, 1,4, alpha 1,4, alpha 1,6. Okay, so you can find some similarity with the amylopectin part of starch. It is also known as animal starch because its structure is similar to amylopectin and is rather more highly branched. Okay, sorry, it is not actually the reason. This should be the separate sentence. It is also known as animal starch because it is storage polysaccharide of animals. Fine. And there is some similarity with the structure of amylopectin. But difference with amylopectin, if you consider, it is even more highly branched compared to amylopectin. So this is the structure, 1,6 and 1,4. So see, in this part you can see 1,4, and where there is branching, there is 1,6 linkage present. Both are alpha. In glycogen, the branching occurs after every 8 to 14 glucose units. That is, if you consider linear chain, suppose there is already 8 to 14 glucose units, and then the branching will start. But in case of amylopectin also there is branching. But in that case, after 24 to 30 units, there is branching. So in this case, the branching is occurring earlier compared to the branching that you have seen in case of amylopectin. On hydrolysis, it will give glucose because it consists of only glucose, and the excessive glucose that is present in the body, that will be stored in the form of glycogen, and it is animal storage polysaccharide. So obviously it will be, glucose will be stored in the form of glycogen. Question related to glycogen. Which of the given statement is incorrect about glycogen? The first statement, option 1, it is straight chain polymer similar to amylose. Is it so? What we have seen in case of glycogen, last slide, there is some branching, right? So this should not be correct. Then it is present in animal cell. This is true. Now this information, though you have not seen in the last slide, but this is also important, that sometimes they are present in some yeast and fungi. Fine. So this is also true. Only alpha linkages are present in the molecule that we have just seen. Here we have 1,4, 1,6, but both are alpha type. So this is also true. So the incorrect statement is only the first statement because there is branching. It is not true. It is not like amylose. There is similarity with amylopectin, not amylose. Complete hydrolysis of cellulose. So remember, the second polysaccharide that we have discussed, that is actually cellulose, and the third one is glycogen. So the first polysaccharide we discussed starch, then cellulose, which is plant polysaccharide, and glycogen, which is animal polysaccharide. So when it is cellulose, if you do hydrolysis, then what units we get? Remember, it consists of only, just have a look in the slide of cellulose. It consists of only glucose. On hydrolysis, cellulose yields beta D glucose. So it is not just glucose. This much information is enough to answer the question. It has to be beta D glucose. Okay. If it is mentioned that L glucose or alpha glucose, it will not be correct. Fine. So it is made of only one type of monosaccharide, which is glucose, and it is beta D type. Now in the options here, it is mentioned fructose, ribose, and 2-glucose, option C and D. Now these two obviously not correct, right? Now among C and D, here alpha beta not mentioned, but D and L, two variety of glucose mentioned, D glucose, L glucose, but L is not correct. It is D glucose, which is the correct option, right? So cellulose is linear chain polysaccharide composed of only beta D glucose unit, and this is the linkage, though the question is not asking anything about linkage, but this is the information, correct information about cellulose, fine, which is plant polysaccharide. Among the following compounds, the incorrect statement is, now here we have statement related to different disaccharide as well as polysaccharide. The first statement is saying cellulose and amylose, they are having 1,4 glycosidic linkage. Now in case of amylose, there is 1,4 glycosidic linkage that we have seen. In cellulose also we have seen there is 1,4 glycosidic linkage, right? So this must be correct option. Correct statement, not correct option, because correct option will be that statement which is wrong. Incorrect statement we have to find. Second statement, maltose and lactose, they have 1,4 glycosidic linkage. So the structure of maltose and lactose, if we see, there is 1,4 glycosidic linkage. This is 1, this is 4, this is 1. So this is also true. Lactose contains beta D galactose and beta D glucose, and sucrose and amylose have 1,2 glycosidic linkage. Now in case of sucrose, the linkage is basically 1,2 type, and if you consider amylose, it is actually 1,4. So this is 1,4, and if you consider the first unit, that is, though it is continuing both sides, but in this case this is carbon number 1. How to know carbon number 1? You just have to check oxygen is attached to this carbon, so this will be carbon number 1. It is actually the aromatic carbon, okay, which is always the carbon number 1. So it is 1,4. So this statement is, it is not completely correct. Sucrose is 1,2, but amylose is not 1,2. So we cannot say that both of them 1,2. So this is not correct, and this is the correct option. And in case of lactose also, this part is galactose and the second part is glucose. Okay, so this is correct. Last statement is false. So that is, it is correct option. The term anomers of glucose, it refers to what? The term anomers of glucose, though this question, as if actually it is based on whatever we have learned about carbohydrate. There is some common question now we are going to discuss. Before this, the questions that you have seen, that is related to the topic. That is a topic that we have just finished. But now, as the whole carbohydrate, it is finished, because next class we will start other topics, which is, that is protein, amino acid. There are some other biomolecules, but carbohydrate as biomolecule, this part is common. And now we are discussing some question which is based on this whole topic, which is carbohydrate. So that is why now we're discussing the term anomer of glucose. It means actually what? Isomers of glucose that differ in configuration at carbon number 1 and carbon number 4. This is the first statement. Then the second statement, mixture of D-glucose and L-lactose. Enantiomer of glucose. And the last statement, isomers of glucose that differ in configuration at carbon number 1. In one class, if you can remember, I have discussed anomers and epimers in detail. So epimers, they are those isomers where all the chiral centers, their configurations are same for these two isomers. Only at one chiral center, the configuration is different. So if this is the case for two isomers, they are epimer to each other. Now anomer is also one type of epimer, but this term is mostly used for sugar molecules. In sugar molecules, when you consider the cyclic structure, just consider the carbon where the cyclization is actually occurred. If you consider that carbon, the newly generated OH, it may be alpha or beta, two types, right? Whether it is below the plane or above the plane. That means only that carbon is deciding whether it is alpha or beta. Rest of the carbon, their chiral centers are same. So it is only carbon number 1 where the newly generated OH is present that decide whether it will be having epimer relationship or not. And anomer is also one type of epimer, but this term is mostly used for sugar molecules. And this carbon where the carbon where this newly generated OH is present, that is actually known as anomeric carbon, and the two isomers, they are known as anomers. So obviously the first option is not correct. That is isomers of glucose that differ in configuration at 1 and 4. Obviously this is not true, and it is not mixture of D-glucose and lactose. This is also not correct, but the last option is basically correct option. Okay, so anomers, they are actually a type of diastereomer. Though I have said that epimer, if you consider the definition of it, it is two isomer where they are differing from each other only at one chiral center. Rest of the other chiral centers, there is actually no change. They are absolutely same. So if you see these two structure, one is alpha D glucopyranose and another beta D glucopyranose. Only at this position there you can find some difference. So here when it is alpha, which is below the plane, this is a newly generated OH, because if you consider the open chain structure, it will be like this. So if I draw it, OH, H, H and OH. So here carbon number 1 is that carbon where this OH, which is in blue color, that is the newly generated OH. If this oxygen, it is attacking here, this is a newly generated OH like this. So depending on the configuration only in this position, because rest of the part, that is carbon number 5, 4, 3, 2, these 4 carbons, they are actually chiral center. Carbon number 6 is not chiral center. Just compare with the beta D glucose 5, 4, 3, 2. So all these 4 carbons, their configurations, they are same. But if you look at carbon number 1, now OH is below, here it is up. So only at one position there is different configuration. That is the definition of epimer. But why we are using the term anomer? Because anomer is used for the structure, that is for carbohydrate structure or sugar type of molecules. And epimer, that is actually a type of diastereomer also, which is in broader sense if you consider. Fine. There is enantiomer, there is diastereomer. So diastereomer, they are also stereoisomer, just like enantiomer. Enantiomer is also mentioned in option 3. But when it is enantiomer, the two isomer, there will be some mirror image relationship. But for diastereomer, there is no mirror image relationship. So the broader term here is diastereomer. So epimer is one type of diastereomer. But for carbohydrate molecule, we use the term anomer, which is a special case of epimer. It is a cyclic form of sugars or similar type of molecules. That is any carbohydrate molecule. It may be non-sugar also, differing in the configuration at the anomeric carbon. So carbon number 1, where the OH is generated because of this type of mechanism, that carbon is known as anomeric carbon. So for glucose, it is C1 atom of aldose, because glucose is aldose. Aldose means where aldehyde group is present. But there is another type of sugar also which is containing C double bond O. So for ketose, you can consider the structure of fructose. So though here the question is related to glucose, but I'm giving some extra information that for fructose also, now there is also anomeric carbon generation possible, but the anomeric carbon now it will be at carbon number 2. Example is fructose, because fructose is ketose. Remember, if you consider the structure of fructose, I am just writing the first 2 carbon. This is the first carbon for fructose because there is no free aldehyde group. Now the second carbon is C double bond O. Then there are some other carbons. But now the newly generated OH, that will be generated at carbon number 2. It will not be generated at carbon number 1. So that is why when it is aldose, anomeric carbon is carbon number 1. When it is ketose, anomeric carbon is C2, because now the carbonyl, it is at 2 position, 2 ketones. So that is why anomeric carbon, it may differ depending on whether it is aldose or ketose. So the term anomer of in case of glucose, it is because of the isomers of glucose that differ in the configuration, which is at carbon number 1, because the other carbons, I mean 3, 4, 5 and 2. Here chiral centers are 1, 2, 3, 4, 5. But 5, 4, 3, 2, these two, 5, 4, 3, 2, their configuration is same. Only carbon number 1, it is different. So that is why. What should be the correct option? This should be the correct option. Okay. So this question, it is based on the topic which is anomers. So though we have some time here, but I will not start the next topics which is completely different. I mean amino acid proteins, that we'll discuss in the next class. So this chapter is biomolecules, and when it is biomolecules, first biomolecule we have in the syllabus, that is carbohydrate. Okay, so that part is finished. Next day we'll start some other biomolecules that are present in the syllabus. Okay, so we are at the end of the session. Thank you for listening.